Hi everyone, welcome back. So up until now, we've mainly talked about living microorganisms. Bacteria mostly, but also fungi, archaea, plants, even animals and protozoans. These we've mainly explored thus far in the context of pathogens, things that actively cause disease. But it's important to remember that not all pathogens are alive. There are lots of things, like viruses and prions, for example, which are infectious proteins, that are not actually alive. They're not considered living things, but they still have the ability to cause disease. And in their ability to cause disease, we see some interesting things. These, what we call infectious particles, behave a lot like a living thing, despite not actually being alive. So we're going to focus here first on viruses, and then in later units we're going to talk about prions. Viruses are not actually alive, despite them doing several things that we often associate with living things, like having DNA or RNA, having some kind of genetic information, evolving over time, and reproducing, making more of their species, of their particular variant. But they don't do some really important things that living things collectively all do, or they don't have certain features that are uniform for all living things. For example, viruses are not made of cells. As we previously explored, cells are the basis of life. Viruses literally are not made of cells, and so they cannot be a living thing by our current definition of what is alive. But they also fail to do a bunch of other things. They don't grow or develop over time. They are made, and then they remain the same up until they infect something new. They don't have the ability to maintain homeostasis. There's no work that a virus does. It doesn't take and use ATP to do metabolic jobs. It's all about getting the cell that it infects to do all of the things that a virus does for it. Likewise, it doesn't really eat, and it doesn't generate energy. It doesn't do respiration. It doesn't do any real metabolism on its own. All of the things that it does using the resources that a cell has. And so because of this, viruses are not considered to be alive and are not considered to be organisms. Similarly, prions, these misshapen proteins, are proteins and not cells, and also fail to do a lot of these things that viruses also fail to do. And so we don't consider them to be living things either. That doesn't mean that they aren't an effective pathogen, of course. They are. So in terms of size, this is kind of what we're looking at in terms of how large and small these things are. So here we have a regular red blood cell, human red blood cell, which is about 10,000 nanometers in diameter. Smaller than that are a typical bacterium like E. coli, 1,000 to 3,000 nanometers. So very small. But smaller than that, still, we have an assortment of viruses. So here we have the smallpox virus, a tobacco mosaic virus, which is the first virus that was ever discovered. We have bacteriophages and things like that, a polio virus over here, which are significantly smaller than a typical bacterial cell, and orders of magnitude smaller than a very small human cell. Now let's talk a little bit about viral anatomy. So viruses come in two different main configurations. A virus can be naked, as in it doesn't have an outer lipid membrane that covers the majority of the virus. Or it could be enveloped, meaning that it has that membrane on the outside of it. So a naked virus is composed of two things at its simplest. We have a capsid, which is a protein shell that encompasses the virus, and then the nucleic acid, the genome that sits inside of that capsid. Okay? So the capsid is made of a bunch of small subunits called capsomeres that collectively come together and build the virus. Sometimes they take on somewhat geometric shapes like this. And then the nucleic acid comes in a few different varieties. It could be DNA, like us, or it could have RNA, which is just a single-stranded nucleic acid instead of DNA, which is double-stranded. So this is really just the code that the virus has to make more of those viruses. Now, these enveloped viruses are naked viruses, but they have an additional wrapping. They have this lipid membrane. And the really interesting thing about this lipid membrane is that it doesn't belong technically to the virus. The virus didn't make this membrane. Instead, it captured the membrane from the cell that it infected. And as it leaves the cell, as it does this budding process, it splits off and steals a large part of the membrane with it. These enveloped viruses will contain not just the envelope on the outside of the virus, but the envelope will also contain these special viral glycoproteins that we call spikes. So these are different kinds of features, mainly proteins or glycoproteins, that adorn the outside enveloped viruses. And these are essentially ways that the virus is going to interface with the potential host, trying to get it to be tricked into taking it up. So we can take these two categories of viruses and we can divide them further. We can divide them by the type of nucleic acid that they have inside them. As I mentioned before, some viruses have DNA for their genome. Their information is stored as DNA instead of something else. Other viruses use RNA. So we categorize these as DNA viruses or RNA viruses, respectively. Now, viruses kind of get a little bit weird with this because sometimes a virus will change the kind of DNA or the kind of RNA that it uses. Normally, we encounter DNA as a double-stranded molecule. You'll often be familiar with that natural double helix structure that we often see in like popular media when we talk about and hear about DNA. And then RNA is a single strand instead of two strands. So you can label that double-stranded like DS or single-stranded SS. And this is normally what we see for life broadly. Living things really just have either double-stranded DNA and single-stranded RNA when they use them in their bodies. Viruses, though, kind of get really experimental with this. There are some examples of viruses that can be double-stranded RNA or single-stranded DNA. So things get a little bit weird, but generally speaking, viruses tend to be double-stranded DNA or single-stranded RNA. Viruses are parasites at its simplest. Their whole job is infecting another host and then utilizing the resources of that host to make more of that virus. So we're going to talk here about the life cycle of a virus. These viruses ultimately require a host to replicate themselves, to make more of themselves and continue their lineage. If they don't get a host to take them up and make more of those viruses, there is no perpetuation of the virus. Which is really interesting and good for us because, in theory, if we could stop organisms from being infected by a virus, we could eradicate that virus. Many of these viruses are what we call host-specific. It just means that they only really target or are capable of effectively infecting one or a couple different species or a very limited group of organisms, typically pretty related organisms. So for example, HIV is a type of virus that really only infects humans and certain primates. And primates are our close relatives. They can't infect like whales. They can't infect trees. There's a bunch of things that they can't infect. Only humans and only primates are capable of being infected. This host specificity comes mainly from the evolution of these viruses as they become more and more effective at being a pathogen. So pathogens that are successful at replicating and spreading to new hosts are the ones that get passed on. They are the ones that continue their lineage. Any viruses that are not successful at replicating themselves don't get to make more of themselves, and so they disappear from the population. Viruses today are perpetually moving towards being effective as pathogens for specific hosts. That doesn't mean that those hosts can't change, that they can't maybe acquire a new host, but this is the general trend and why we tend to see a lot of host specificity. Now, this force is opposed by the host as well, because the host is a living thing that can also evolve. So these organisms that it infects are also perpetually changing. They're perpetually evolving in hopes of being more effective at avoiding that infection or overcoming that infection. And so there's a back and forth that often develops here. So these strategies for becoming a better and better pathogen often involve either avoiding the immune system or subverting the immune system when it becomes active. Specificity goes beyond the host. Even within those specific hosts, there can be a lot of specificity. For example, for HIV, there are certain cell types that HIV can infect and not others. HIV mainly infects the helper T lymphocytes. These are specific immune cells, and while HIV can infect a couple other types of cells, most of the cells in your body cannot be infected by HIV. So your muscle fibers, your osteocytes, your neurons, these are cells that are not impacted by HIV because the virus literally cannot infect them. It is specific not just to species, but to cell type as well. And this is something that we see pretty broadly with other viruses, where even other viruses that infect other host species can't infect every cell in your body. They can't infect every cell type in your body. Let's talk a little bit about the viral life cycle. So viruses are really diverse in who they infect and how, but all viruses have to do basically the same cycle. There can be variation in how this cycle plays out, but it generally follows this pattern. First, the virus has to be taken up by the cell or have its genetic material taken up by the cell. It has to bypass the initial defenses of the cell, like, for example, the cell membrane or potentially a cell wall. The virus then has to utilize the tools of the cell, the various resources and enzymes and other materials that are inside of a cell, to replicate itself. This means hijacking all of these resources, the different enzymes that the cell has, and making them now make the pieces of a virus. The capsid, those capsomeres, replicating the DNA or RNA of the virus, replicating whatever specific enzymes might be within the virus as well, any of those spike proteins, things like that. And along the way, it also has to avoid detection by the cell or body's immune system and avoid being killed by it if that immune system is active. Then, at the end of all of this, the virus has to escape the cell to find a new potential host. It needs to go and continue that process so it can go and find new hosts to make more and more and more and more and more. That's ultimately all that a virus does. There can be a lot of different ways that a virus escapes a cell. Sometimes that is through destroying the cell or lysing it, so they burst it apart and dump their contents into the environment, but there can be other ways as well. So, a viral infection strategy, these different ways that these viruses will actually conduct their infection can vary considerably. So in viruses that infect animals, we can see a lot of different strategies that involve getting past the cell membrane, including these different envelope strategies. So remember that some viruses are envelope viruses. They contain stolen cell membranes. They can use those cell membranes to basically fuse with the existing host membrane. They can use that to be taken up by the cell, or they can be swallowed by the cell as well. So they can actually they can trick the cell into thinking that it's a food or some other kind of resource. and bring the virus into the cell that way. Doing so results in the virus being broken down or broken apart, and ultimately releasing the inner contents, that genetic material. The cell is then tricked into producing all these different proteins that the virus is made of, and then assembling those viral particles as well. In the end, you end up with virions. A virion is just a fully assembled viral particle. On assembly, these new virions are released steadily over time, or sometimes very catastrophically through cell lysis, where the cell just literally is torn apart. There are so many little virions inside of the cell that it swells and bloats up and then is torn apart, leaking all these new virions into the environment. So this is kind of how this looks. In one example, a naked virus can do direct penetration, where it attaches to different receptors on the cell membrane, and the viral genome is deposited directly into the cell. So the capsid stays outside, but the viral genome enters. In other examples, you can have envelope viruses and have different ways that they interact. So you could have the lipid membrane fusing with the host. You could also have the cell being swallowed through endocytosis. So the cell can be tricked into thinking that this virus is food when it interacts with the receptors on the outside of the cell. And then the cell can wrap its membrane around that virus and then bring it inside, essentially swallowing it, and then create an opportunity where that membrane can break apart and spill the contents into the cell. Then you have really interesting ways of viral escape. So in this case, we have viruses, when they go to escape, they actually drag the cell membrane with them. As the virus begins to leave, the virus can pinch off some of that envelope with it and take it as it leaves. So now you have an envelope virion leaving the cell. This is that example of budding that I talked about before. Then we have another major group of bacteria that I want to definitely talk about. It's very important. Bacteriophages. So a bacteriophage is a really abundant type of virus that only infects bacteria. These little infectious particles make up a huge percentage of bacterial mortality, far more than things like antibiotics and competition among other bacteria. We have viral interactions, viral infections, particularly in places like the ocean and other kinds of marine environments. This can make up to 70% of bacterial mortality. So it's a major source of bacterial death. Now, across the earth, we have an estimated 1 trillion bacteriophages for every grain of sand in the world. Their abundance is staggering. They are extremely widespread. They do not, as a reminder, infect humans or animals or plants or fungi or anyone else. They are bacteria specialists. So, bacteriophages are really interesting, and they have a certain life cycle, and they can replicate themselves with 2 main methods, and these methods can interact and overlap. So, bacteriophage life cycles can start with the lytic cycle. Okay, this is a more immediate kind of replication where we have a viral particle that attaches to a cell and then injects its genome into the cell. It has these mechanisms that allow it it doesn't have to be swallowed by the cell. It can just literally attach to the surface of the cell and then puncture the cell and insert its genome. The bacterial cell will then transcribe and translate the proteins that are encoded in the bacteriophage genome. It'll be forced to make all of the parts of that virus. And then it will replicate its genome. It'll make everything. Then those viral pieces assemble into new virions. In the end, this cell has been turned into a viral factory. And it has replicated so many of these little virions that the cell begins to inflate, it begins to break apart, and eventually a rupture occurs that spills the virions into the environment. Now, all those little virions can go out into the wilds, they can go and find new bacterial cells and continue their infection. This method, it is very rapid replication for the virus, and it can lead to the quick death of the host cell. Then you have the lysogenic phase. So the lysogenic phase is a more long-term form of replication. This is where we have the bacteriophage still inserting its genome into the bacterium. That genome, instead of being immediately used to make new virions, it starts to insert itself into the host genome. It literally will stitch itself into the genome of that bacterial host. Now, that cell is a hybrid. It has DNA that is both bacterial and viral. And as it replicates, as that cell goes about its life, it will do things as normal without expressing those genes. This happens because those genes that came from the virus are temporarily silenced. They're shut off so they are not being read and made into the different proteins that the virus normally makes the cell make. Eventually, though, as that cell continues to replicate and make more of itself and more and more and more, each one of those new cells carrying with it the viral genome, some of those cells can reactivate those silenced genes. Eventually, different kinds of environmental triggers could stress the cell, could make it change the way that it hides or activates certain genes. And as a result, those viral genes can become unsilenced. They can be activated again. By this time, a lot of cells have likely been produced that have these viral genes. So this is a way that the virus can replicate itself without actually making more of the virions. It can spread its influence without having literally more viruses. Once activated, though, the cell will switch from the lysogenic cycle into the lytic cycle, into that period of viral replication, the one that turns the cell into a viral factory and eventually results in the cell tearing itself apart, lysing, and then dying. This is what this looks like, okay? So here up top, we have the lytic cycle. We have the bacteriophage inserting its DNA into the host. Now, this phage DNA is going to be read by the host cell and used to replicate more of these virions. So the materials are built by the cell, the DNA is replicated by the cell, and everything starts to package itself back together. Eventually, there are so many of these viral particles that the cell is torn apart from the inside and all these contents spill out into the wild. However, the lysogenic cycle could be the starting point as well. And here we have the viral DNA being inserted like normal, but instead of staying independent, instead of being independently read and used to make viral particles, it attaches to and inserts itself into the host genome. As it does, that cell can replicate and replicate and replicate and replicate, spreading that genome alongside its own. Eventually, though, under typically stressful conditions for the bacteria, that silenced genome could be activated. It can be used to create a separate genome once again. It separates itself from that host and then can use that point to enter into the later points of the lytic cycle. Again, turning this cell into a viral factory that then makes so many of these bacteriophages that the cell bursts.